Visual thinking and different modes of observation can help us to really connect to anything that we're looking at. In my case, it's probably an underrepresented organism. I think of myself as a visual artist more than anything else, but I have a special interest in biological subject matter. But I approach my subject matter kind of like a science illustrator. This means that I really try to understand what I'm drawing and to share that understanding. Curiosity will push me in for a closer look. And a science illustrator will put clarity above all else. More important than photorealistic lighting effects is illuminating all parts of the subject. Here we see a native oak leaf grasshopper adorning the first non-photographic cover of this journal. My next assignment was a full-page illustration to accompany a Gary Nabhan article about helping migratory pollinators. Here, instead of just restating the author's insights, I wanted to expand on what the author had to say. I wanted to provide more information. And I thought that I could do this with the language of images. So I chose one of the characters from that article and decided to offer a closer look. This is our apex pollinator, the honeybee. And honeybees have a slight static charge on their bodies. So when they land on a flower, pollen literally hops onto them. Kind of like those little annoying pieces of styrofoam that cling to your favorite black shirt, right? It's like the dog hair of the animal world. At the bottom of this illustration here, I have some line drawings that I also included, and these show the different tools built into the honeybee for what I call pollen management. We have things like an eye brush, we have an antenna cleaner, and even a little compactor built into the hind legs for compacting pollen. A science illustrator will often look at lots of samples and different angles to really distill and condense this information into a compact, understandable summary. These are some photographs that I took while visiting an insect collection. So I was just gathering some reference for the last two projects you saw. Now I do prefer to work from observation, and I was able to observe some of these insects, but it was really nice to cut down on the eye strain by having something that was a little larger than half an inch to stare at. And when I take this approach to my illustration work, my work usually starts on paper, and I like to draw in pencil, graphite pencil. When my pencil drawing is all done, I pop it into a flatbed scanner, and this digitizes the print for me, or the drawing for me. I'm kind of a light-handed drawer, so I boost the contrast quite a bit, and then I use a digital drawing stylus and little tablet to digitally paint in layers of color. So the end result is a landscape of textures and details living underneath a translucent skin of color. And in this particular project, there was a little money left in the printing budget for a custom blue ink and a really cool thing we call a spot gloss, where we just apply a high varnish to one part of the cover. So I chose the eyes and the brain of this moth. To some tech-savvy artists, working digitally might make the most sense from start to finish. For me, I prefer to do my thinking on a paper playground. Here we can see a few examples from my sketchbook. I think of drawing a lot like writing, in that it's a way to put your thoughts onto a surface that you can actually look at. And it's a really nice way to visualize what you're thinking and organize your thoughts. So whether I'm at a natural history museum or an aquarium, or maybe an animal refuge, I prefer to draw from live observation whenever possible, rather than using a photograph that's already been flattened two-dimensionally. And I find that this mode of drawing, this unmediated form of observation, really feels like the most human way to experience an object or a living subject. It helps me to better understand how that thing occupies three-dimensional space, and it also can help me understand how it moves through four-dimensional space when there's time and movement involved. So these are a mixture of pencil, and there's also some dirt cheap ballpoint pen in there as well. It's a pretty portable combination. And nobody tries to steal your fancy pens either. So I think ballpoint is a really wonderful material. I think it's a little bit underrated. I carried live observation and ballpoint pen into this project that I called Groundward. And I created this as the first artist in residence at a riparian preserve just north of Tucson. Groundward is a collection of 10 observational studies of ant nest openings that survived summer monsoons of the Sonoran Desert long enough into the summer to be drawn and remembered. So if you want to get touchy-feely about it, this is a way for me to document my kind of quiet time and space that I shared with some ants. No uncles. So again, these are ballpoint pen drawings. Below each of these, I have some little field notes, mostly observational stuff, things that I was hearing, like thunder rumbling over the mountain, things I was smelling, like that ozone, like creosote resin smell when the first raindrops touched the earth. And as I was drawing these, I tried to also eliminate as many variables as I could to really create a uniform perspective for all these. Eliminating unnecessary variables is another science illustration tactic. So, to that end, I made sure that I observed all of these from about 2 feet away, sat on a little stool. I drew all of them at life size, so on average about 6 by 6 inches. And I figured out where north was. I figured out the north side of each of these little ant holes and aligned that with due north on a compass. All right, so back to those little notes again at the bottom. Aside from some little musings and ruminations, you also see the number of the ant nest, so I could revisit it on my little map that I drew. And you will also see the species of ant. I figured out what type of ants were making these. You'll see the time and the date, and you'll also see down there a set of geographic coordinates. And that was just to help pinpoint the site of this very temporary architecture. Now, in drawing these, I employed another science illustration tactic. even though this was kind of more of a fine artsy project. I had to sift through lots of visual noise and then figure out parts that I wanted to emphasize. Okay, sifting through the noise and finding the important bits. I had taken a few reference photos quickly of these, just in case the rains came early, or in case a clumsy naturalist stepped on one or something, which I didn't do. But these photos were awful. They were a terrible reference. They didn't convey depth well at all. The texture in them was overwhelming. Everything was in sand, and I'm under the bright desert sun, trying to look at little details inside of a minuscule cavern. It was just really hard to see what I was looking at. But the human eye and also a pair of sunglasses really saved the day, because the human eye is a dynamic aperture, so changing contrasts quickly isn't a problem. And maybe more importantly, the human eye can really shift its attention to different landmarks that help define form. So while we're staring at the ground, looking groundward, if you will, why don't we dig a little deeper beyond the parched surface? An organization called the Soil Food Web School approached me to create their official artwork for an international conference about regenerative farming and agriculture, and by extension, biodiversity in soil. So I did some research, visual research, and some reading, and figured out some of the beneficial microorganisms that this organization wanted me to illustrate. And I ran my sketches by the client, and after getting approval, I felt comfortable moving on, which for me means taking a closer look. Now these things are really tiny. You can see some universal scale bars on these. Reading, if you see a weird symbol there, that's very, very, very tiny. It's much smaller than a centimeter. We have micrometers, we have nanometers, all kinds of tiny measurements for these minuscule beasts. So I had to look at microscopy or microscope images. And I actually looked at two different types of microscope images. Here you can see something that's probably more familiar to you, optical microscopy, whose colorful subjects display a backlit transparency, because the image is made by light passing through these things. Okay? When I was a kid, I tried to look through a carrot slice or something, and I realized I had to make that thing like as thin as a sheet of paper so I could actually, see through it, right? It's optical microscopy. These have that really nice transparency, full color. The downside is that really shallow depth of field. And that means that whatever we're looking at comes into focus as a really tiny, shallow sliver of space. So for that reason, I also looked at another type of microscopy, scanning electron microscopy, whose subjects don't have any kind of transparency to them. They appear very opaque because they're being bombarded by electrons rather than visible light. But the upshot is that the images produced by this process have, I guess you could say, a really incredible depth of field. It's also worth noting that electron microscopes and our most powerful deep space telescopes gather data and produce grayscale images that require color to be added in afterward by some kind of interpreter, maybe software, maybe a human interpreter. Color aside, my goal here was ultimately to visually strip away all of the soil in a sphere-shaped sample of beneficial microfauna. And these really charismatic buggers are all swirling around, having a little party around some fungus-enhanced root tips. So I guess you could say that I tried to make up for the shortcomings of each type of microscope by combining the best of both worlds in my drawing. I also leaned on some electron microscope reference in drawing these adorable bug heads. I mean that, they're adorable. A science center needed some really generalized insect heads to showcase 4 main evolutionary pathways that gave rise to the main categories of insect mouthparts. That was a mouthful. That was mouthful. So we have, I guess you could say, sponging mouthparts or lapping mouthparts on a generalized fly. We have chewing mandibles on a generalized ant. We have siphoning mouthparts on this little mothy fellow. And everybody's favorite, piercing mouthparts on a mosquito. I imagined all of these and kind of recombined them with my reference and in my head, all at the same angle to really get rid of that extra variable when trying to draw a comparison between all of these. Also, I applied a really muted color to the heads to downplay the changes in shape there, or the different head shapes, but used this really bright blue color to highlight the variables we're supposed to look at, the mouth parts. The museum director came to me again and said, hey, can you also create a big welcoming presence for the entrance to the entire insect exhibit? Bless you. And this would be, I guess we would kind of refer to this thing as the welcome bug. Needs a theme song or something, I don't know. So this would become a 2-sided illustration that would stand 5 feet off of the ground. And I was given quite a bit of freedom in actually choosing what to draw for this. Bounced some ideas off of the director, and I arrived at a type of beetle. And to be specific, this is a type of jewel scarab. Okay, don't ask them about rolling dung balls. They don't want to hear about it. I chose this in part, for one thing, it doesn't have big scary jaws or something. But I also really like the white gold and brushed copper paint job. Now, if I wanted to live a simple, easy life, I would have drawn this head on with maybe kind of a symmetrical point of view or a symmetrical stance. But instead, I proposed something that was a lot more challenging to pull off, and that was building some personality into the stance of this beetle, having some asymmetrical movement, and also trying to achieve a little bit of 3-dimensional depth and tilt in there as well. That was especially difficult because these 2 illustrations had to have the exact same silhouette since they were on either side of a cutout. So after lots of sketching and hours of staring at this little beetle on my desk, woo, Friday, I feel like I arrived at pretty accurate anatomy and was able to combine a real natural range of movement in the joints with a really healthy dose of humanity, in part with this, this kind of human stance. So I've addressed several science illustration tools and tactics today: clarifying, condensing, sifting to emphasize, opening up inaccessible spaces, and even adding some approachable humanity. I feel that all of these really come together in a culmination in my final example today, and also bring a few more little tactics to the table. This is symbionts. Symbionts was a personal project, so another project that wasn't commissioned. And it had a three-dimensional half, three-dimensional sculptural component, and the other half was two-dimensional illustrations. So with the topic today, I'm going to focus on the two-dimensional half. And symbionts, the title I chose for it, comes from the word symbiosis, which is the living together of unlike organisms. So I've got 5 different symbioses in the ocean here, 5 different pairs of symbiotic organisms. And some of them are parasitic, right? That means we have one organism taking from another without permission either, taking from another without killing the host. Some of these are a little happier. Mutualism, that's when both organisms benefit from the symbiosis. So we'll start things off with parasitism, because it's about dinner time and I think we deserve it. I give you the tongue-eating louse and the red snapper. The tongue-eating louse is a pill bug-like isopod, and when it's small, it swims in through the fish's gills and latches onto the tongue. From there, it getting kind of queasy just talking about it. From there, it feeds and grows on this necessary organ. Yes, fish do have tongues. Feeds on this necessary organ until the tongue actually shrinks away. And at that point, the louse itself replaces the tongue. I'll draw it, just don't make me touch it. Beneath the full color artwork, I have some diagrams that I drew that I feel visually tell this story. And I'm also using yet another science illustration tool here. We call this the cutaway view, which affords us a look at the inside of something while simultaneously showing the outside as a point of reference. Let's do another parasitism, because I can hear the crowd loves it. So we have another pair here. The larger of the two is a Portuguese man-of-war. And that's basically a fused colony of little simple gelatinous animals all floating together, kind of like a pirate ship. The largest of these is gas-filled, and that keeps the organism afloat. And there's also a little sail, so it catches the wind like a sailboat. There's no motor or anything involved. All right, so this thing just floats along on the surface, passively catching any clumsy fish in its long, stinging tentacles, which can get much longer than what you see here. These stinging tentacles are loaded with thousands of specialized stinging cells. Each of these is spring-loaded and contains a venom-soaked harpoon that fires upon contact. And that can be really bad news when you have hundreds or thousands of these things firing into you at once. But our parasite, the blue dragon sea slug, swallows air, floats upside down on the surface to feed on the tentacles. And it can do this without triggering the stinging cells, without setting off these little harpoons. In fact, it's so specialized at this type of feeding that its body can pass these stolen weapons into its own appendages for its own self-defense. But enough with these cunning parasites. It makes me think of politics for some reason. Why don't we wrap up with a mutualism in the name of teamwork and collaboration? The golf ball-sized bobtail squid buries itself in the sand during the day to get some shut-eye, to sleep. At night, this little squid comes up, goes for a swim to hunt. But so do its predators. To a predator swimming underneath this little squid, a little silhouette against the moonlit surface would make for a very easy target. So there's a workaround. Nature is just one big arms race. The squid has a specialized organ, and it looks like this when the squid is a juvenile. And this organ's job is to first take in seawater, and then it isolates just one specific strain of bacteria from that sample. It then cultivates that bacteria. And it does this because that specific type of bacteria glows in the dark when it's fed. So for the rest of the squid's life, this organ has a pretty simple job. It just has to provide a home and tasty food for this colony of bacteria. Okay, it's a mutualism, so there's got to be some kind of trade-off, right, or a payout. So in return for a home and for food, the bacteria gets light. The bacteria produces light which hides its silhouette against the moonlit surface. As the squid gets a little bit older, those appendages on the organ shrink away, and we see the mature organ here as just two lobes, kind of like beans or something. Now, as if all of that wasn't incredible enough, there's more. Beneath this light organ is the ink sac, which this species of squid can contract and expand. And it acts basically like a dimmer switch. That means that the squid can control the amount of light coming out of the organ to match the ambient light of any moon phase. I hear some wows, and I feel the same way. So this is another one of those little illustrations at the bottom of the larger print there. I've boiled away a lot of the extra details and things. You know, we don't have to look at any guts or anything to see what's inside this squid. I've also compressed time, right? I'm showing a process in a sense. And I've also taken out other variables that might cloud the way we interpret the stages of what's a really elegant natural process. And I may have humanized that squid a little bit. I think he's a cutie pie, but I think that's accurate. So I hope that you've found at least something today that's sparked your curiosity enough for your own deeper dive, maybe through drawing, maybe through further reading, maybe a nature documentary, or maybe taking a moment for some really close, focused, thoughtful observation. Curiosity can show us something that we never knew existed. But the more we learn about that thing, a fish, a bug, the humble microbe, the more likely we are to care about it. Thank you.
Thank you, Bobby. It's terrific.
Thank you.
So the first slide and the last slide are of a googly-eyed fish. And I'm guessing a few of you guys have noticed this slide, but Bobby didn't say anything about this fish. We saw a different fish, one that was missing a tongue. We've had a few conversations, and what I've learned from those conversations and from this presentation, at least an appreciation of looking when I see a scientific illustration, what is it that the drawer, the illustrator, what have they observed? What are they trying to accentuate? What do they want us to notice and learn? And so I see this, and I see googly eyes. And that's what pops out to me. So what is it that you found interesting about this? What is it you're trying to share with this image?
Well, this was, just to give a real quick backstory, the museum that asked for those insect heads and the big welcome bug more recently asked me to do some large deep sea illustrations for a permanent display in their ocean exhibit. And so there are 6 of these. This was my favorite one. And yeah, you caught on to the googly eyes. It's kind of hard to miss those, right? They're a little creepy. And I thought it was just kind of a nice choice for something, you know, a presentation about looking and drawing. So that definitely caught my eye first. Something I found even almost more interesting about this is that this type of fish, called a barrel eye fish, actually has its eyes inset inside of its head, and it actually is looking through a cockpit-like forehead. So those green orbs are trying to gather a lot of light down in the deep ocean. They're aimed up at the surface, so it can steal food from jellyfish and things. And it's got this clear cockpit-like head. Yeah, and its catchphrase is, "My eyes are up here," I guess.
All right. Well, thank you.
Well, thanks, Brandon, and thank you, everybody.
Really enjoyed the talk.